All right. Well, why don't we get started, everyone? As a reminder, we are recording this, and this is going to be on the YouTube channel either later today or tomorrow, hopefully. Let me know if you have any problems hearing or seeing anything. So today we're going to talk about single-cell RNA-seq analysis. Before I get into it, it's going to be a little bit more advanced than what we've done in the first 3 weeks. Certainly, I don't expect everyone to be able to, follow along, with every tiny detail of the code. We're not going to be running all the computations that we need, because some of those, even on a pretty powerful computer, can take a while to run. So I have some pre-made data sets that we're going to use for demonstration purposes. And what I'm going to do is I'm going to explain to you sort of the more or less the practical details of how to do the analysis. I'm going to run through the code and highlight the important points. And then we should, not take the whole hour this morning to get through all that. And I would like definitely anyone that's got questions to put them in the chat or in the Q&A as we're going through, and I can clarify as we're going through, because we're going to have a little bit of extra time today, I anticipate. So today we're going to get through essentially the basic processing of the data, and then next week we're going to talk about visualization and some common computations that you're going to want to be doing with your data. Okay, so let's get started. Now let me share my screen again here. Okay, so you might be asking the question, why are we talking about single-cell RNA-seq? There's a lot of things that, we could have chosen to talk about, or you might be analyzing different types of data in your project. That's definitely a fair point. I will tell you that there are some fundamental principles that we're gonna go through in terms of understanding data structures and understanding how to manage data, which are well exemplified by single-cell RNA-seq. And so I think there's going to learn some principles there that are going to be helpful if you're, analyzing mass spec data or genomic data, or whatever the case may be. The other point is, in actual fact, single-cell RNA-seq has become more or less a standard assay, for better or for worse. It's extraordinarily powerful for a number of reasons, but it does have some downsides, namely cost and the informatic challenges that we're trying to work through here. So I think it's important to learn how to analyze your own data, and even if you can't do it, or if it doesn't make sense, all the steps, you can't make them work, you need help with it, that's fine. But you should be able to, once you have a data set sort of processed, you should be able to get out of it the things that you want to get out of it, namely figures and tables with data in it. Okay, so by now it's a very standard assay in the types of biology that I do: stem cell biology, cell biology, immunology, all these things. It's very standard. Compared to bulk RNA-seq, there's a number of advantages. Okay, so you can bioinformatically purify your cells. Well, what do I mean by that? Well, often we'll do, maybe 10 years ago, we would have done a experiment where we, sort cells using a flow cytometer, and then do RNA-seq on the sorted cell populations, a bulk RNA-seq experiment. And this is still, can be a good experiment to do under certain circumstances. But you'll find that, there's often a lot of contaminating cells in the FACS analysis, and they can be different cell types, or they could just be poor quality cells with degraded RNA that are diluting out what you're trying to look at. So single cell RNA-seq allows you to select with essentially, obviously, single cell resolution, the types of cells that you want to look for. And this is incredibly powerful. It allows you to detect unanticipated heterogeneity within your population of interest. So let's say you're looking for, you're doing a drug study and you're treating cells and you're looking for changes in gene expression within a cell type. Well, you're going to be able to identify that with vastly greater resolution compared to a bulk assay, because if it's the same cell type, you don't necessarily know what to sort for in order to identify the cell type. So when you're doing single cell, you can find the cells that have responded to your drug versus the cells that didn't, and you can characterize those cells in great detail without the contaminating cells that didn't respond. So that's very important. Another benefit is the generation of gene modules. Okay, so gene modules, we're going to get to this in a bit, but can be sort of an arbitrary notion. So any group of genes that you might want to analyze together, you could call a gene module. The way I'm referring to it is based on calculations that are done in the Monocle package, which I think are very powerful, where they identify in an unsupervised way groups of genes that are co-regulated. So groups of dozens or maybe hundreds or thousands of genes that travel together in a similar way from cell to cell. And this is a really powerful concept because it tells you about transcriptional states in cells, and it identifies them in a way that is unsupervised. And you didn't pick out, say, a T cell exhaustion pathway or a senescence pathway. These things emerge from the data, and so it's much more powerful. Pseudotime. So pseudotime is something that we're not really going to get into, but this is another concept that can be useful for developmental pathways like hematopoiesis or developmental biology. We're going to talk a little bit later, actually next week, about pseudobulk analysis. So that's an important detail. Multimodal is, it's as it says, it's looking at not just RNA, it's looking at protein or chromatin accessibility. So there's a number of technologies that are out there that can add depth to your analysis that are not available from a standard RNA-seq assay. So I alluded to some of these challenges. What are they? So the number one challenge is how to deal with the heterogeneity. So we're going to define many clusters in our example data set, and you'll find that in your own data sets when you work with them. And so how do you quantify that? How do you characterize that? And how do you get some meaning out of those different cells? And that can be a little bit of a challenge, and it's a different way of thinking about biological data than most people have thought of before. So we're going to address these challenges next week, but it's about identifying what your comparisons are. Okay. There are biological and technical barriers. Okay, so it's obviously I think it's fascinating that we can get RNA from single cells, but still there's not 100% efficiency in that, and so you're going to be missing genes from certain cells. And so there's a limit of detection, and so you have to understand the consequences of gene dropout. So if a typical cell expresses, say, 10,000 genes or 9,000 genes, you might only detect between 2,000 and 3,000 genes in a single cell assay. So how does that sort of bias your results in any way? So you have to think about that. computational technical barriers. A lot of this has been mitigated by the 10x company, 10x Genomics. They've developed a cloud computing platform, which is very useful for doing the most computationally intensive tasks, which are the processing of the sequencing, the raw sequencing data. So that more or less has been, largely resolved, but still it can be a challenge, when you're looking at thousands of cells. It occupies, maybe 8 gigabytes, 12 gigabytes of memory. So you need to have a computer with some substantial memory capacity and also processing speed in order to do some of the downstream analyses. And that might be a limitation for some people. Obviously, the coding, the management of the data, these are things that we're going to be talking about, for the next 3 weeks to hopefully help with those issues. Cost is a big thing. These assays are very expensive. The sequencing is very expensive. And right now there's, no great ways around that. There are some ways to multiplex and get more for your money, but it's still an expensive assay. So, I think that's just part of the how biology has transitioned over the last 7 or 8 years. There are alternative methodologies, bulk RNA sequencing, single cell, more or less plate-based methods where you isolate single cells and do, traditional library processing, or more or less traditional library processing on those. These are options under different circumstances, but I think, by and large, over the last several years, the 10x system has essentially taken over and covers a wide range of situations that you're going to find in your experiments. Okay, so how are we going to get these data? So, typically how these experiments go is, you'll run the assay, or you'll collect your cells, and you'll, usually will hand off your cells to a core facility that's going to run the assay. They're going to get sequencing data. And they may that may be just where it ends, and you'll need to find someone to analyze your data, or you'll need to do it yourself. And so fortunately, like I mentioned, the 10x Genomics Cloud allows you really to do this yourself, essentially from a laptop with an internet connection. So let me show you how I do this on a day-to-day basis when I'm working on these. And so this is the 10x Genomics Cloud here, and you can see I've got a number of different projects over here on the left, but let's focus on the if you wanted to create a new project, you can do that from over here. So it's very easy to work with. And so all you do is you click on this thing here to get to your FASTQ files. Yeah, actually, let's go back here. There we go. That's what we wanted. FASTQ, this thing here. There we go. Yeah, so there's the FASTQ files, and what you're going to do is you're going to click on this guy here, and you can browse like on a regular file explorer menu, and then find your cells, find your sequencing files, and then you just upload them. The other way you can do that is through a nice program here, the command line program. That's actually going to work probably faster. If you're able to do that, there's very easy to follow instructions here. So, this is the way to go. You upload your sequencing here, and then what you do, these are showing as expired files here, but normally what you would do is you would select and identify what files, what these files are, what assay was run on them. And then you select them, and then you click on this thing here, create new analysis, and that gives you, it's going to run for maybe 20 minutes or so, and you're going to get, each sample is going to generate an analysis for you. And we can just dive into one of these here. Now, this is a set of basically publicly available data that comes from 10x that I analyzed for the class. So what you get out of this is basically everything that you need to analyze downstream. Okay. So you can select all these files here, and then you're going to download these files. And if I can just go over here for a second, this is, I want to show you what you get out of these. Okay. So if I go here, this is essentially those files downloaded. And the core things that you're going to be getting out of this assay are three things, and they are here: the count matrix, the features, which is the list of genes that you've aligned your data to, and the cell barcodes that were identified. All right. So those are the main data. You're also going to get a very helpful quality control file. And this, you can just click on, it opens up in your browser. It tells you how many cells were identified, important sequencing metrics, how many reads per cell that you got, how many genes were identified. And so this is all produced for you by the program. It's very helpful. Okay. So as I mentioned, use the 10x Genomics Cloud. You upload your FASTQs. You select the pipeline, the analysis pipeline that you're going to use. There's reference genomes for mouse and human in there. There's also xenograft references in there if you are doing a xenograft experiment, so they can align to both human and mouse genomes. And then it will identify which species your cells are from. There's some helpful options that you can select, to either make or don't make a loop file. That's going to give you some interactive ability to interactively browse through your data. I showed you it generates the sequencing metrics. It generates the gene and cell barcode matrices, which I'm going to go through in a minute. You download it. I usually download it with a command line interface, but you can also download it over the web, more or less. It's fast and doesn't use your computer resources. It's also free, by the way, so that's another thing. Yeah, and so there's it will give you some nice warnings as well. Like if you're, you didn't get enough sequencing, enough reads per cell, it will warn you that. Okay, so I showed you very briefly what the data is that you get out of this, and as I mentioned, it's a gene barcode matrix. And so what is this? And I think it's helpful to discuss this in the context of the data structure that you're going to be using in R. So the data structure in R is a complete parallel of those three files that I showed you: the barcodes, the features, and the matrix. The data structure in R is what we call an S4 data structure. If you ever see the word S4, it might be confusing. But the way you want to think about it is, it's like a list. It's a structure of data. It's a flexible. It's a structure of data, but it's not as flexible as a list. There's rules attached to it, and these rules are meant to validate the data object. So there's certain things that are important. For example, as I show you here, you have your matrix, and the matrix is going to consist of columns, and the column is going to represent the cell. So each cell is going to have a column in this matrix, and each gene will be on a row. Now the values in here will be the counts of each gene in each cell. So it's a large numeric matrix. A matrix is like a two-dimensional vector. It's a two-dimensional array of values of the same type. Okay, like a vector is a one-dimensional array of values of the same type of matrix is a two-dimensional array. And so the rules here, the most important rule is that you have metadata, which is attached to this count matrix, okay? The metadata is what tells you additional information about each cell. So think of all the things that you can use to describe a cell. You might want to say what sample it came from. You might want to attach some information about that sample. So what experimental group it was, what replicate it was, time points, treatments, all these sorts of things are features that you can attach and information that you can attach to a cell. Well, it's important that you have the same cells here in your metadata that you have in the count matrix. Otherwise, it doesn't make sense. And they have to be in the same order so they don't get sort of separated from each other and mixed up. So what the S4 data structure does is it validates all that and makes sure everything's all together and in the same order. So that's the basic on the cells and the cell metadata. Well, we can also think about metadata about genes. So, each gene in the reference, there may be 15,000, 20,000 genes, depending upon the reference that you use. Each gene might have a gene name like beta-actin. It has a gene identifier like an ensemble number, and it might have additional features that you want to add to it. So let's say a gene module, or let's say a pathway that you pull out of the literature. You could identify those features about genes, and all this becomes basically, it enriches the your data set. Okay. So again, to summarize, you have in the center of your data object, which is in this case a cell called a cell data set. You have the count matrix with details about the cells held in a separate table called the cell metadata, and details about the genes held in a separate table called the gene metadata or the row metadata. Okay. The other thing that is held in the cell data set is reduced dimensions. All right, we're going to talk about how reduced dimensions are calculated in a bit, but what these are a way for us to visualize in our two-dimensional world the complex relationships and multidimensional nature of this large data set. So let's say you have 10,000 cells. You could describe each one of those cells by 15 to 20,000 different dimensions. If each dimension was a gene, that's the largest number of dimensions that you could use to describe those data. Now that's 10,000 times 30,000, and that's a lot. So you want to reduce that, 10,000 times 15,000, you want to reduce that down, and that will give you eventually 2 dimensions, which we're going to then be able to plot. And these are the UMAP dimensions that you've probably seen a lot. All right, and here's a question. So in matrices, is any point is a single cell? Any column is a single cell. So let me show you on the in R here what I mean. So let's go here. So when you, and these notes are actually going to be available for you guys to look at. So I've already run all these things, so I don't need to run it again. So let's look at the example CDS or cell data set. So when I run that, it says, so we'll just clear this and I'll run it again. So the vignette CDS or cell data set has these dimensions. So it's got 36,000 genes in this case, and it's got 1160 cells. All right. And so as I mentioned, the core bit of this data structure is the matrix, and you're asking about the matrix. So this is what the matrix is, all right? And you can see here, so this is a 36,000 row by 1160 column sparse matrix, all right? And each row here has this ENSG identifier. So what's ENSG? That is the ensemble identifier for one of the 36,000 genes. And it says each column here, so we're suppressing column names, but basically the column names are these things here. This is the cell barcode, and this is the sample that that cell came from, okay? So here, each column here of dots is a cell. So you can imagine that this matrix is 1,100 columns wide. Now, you can see there's a lot of dots here. So what do these dots mean? The dots are placeholders for zeros. Okay, so a sparse matrix, when there's a lot of zeros, there's no point in saving all those zeros. They take up space. We can essentially write them off. And so for the visual representation, they're represented by a point. But when you see a lot of these dots, that's what that is. It's called a sparse matrix. Hopefully that answers your question. So that's the core basically bit of all this. Now I mentioned about cell metadata. Okay, so how do we find out the things that we know about these cells? There's a command called call data, okay, that we can run, and that is going to pull out the table of cell metadata. As you can see here, it's a data frame with 1160 rows and 16 columns. So I can put as many columns on there as I want, but the number of rows is always going to equal the number of columns in the matrix. Okay, so that's the validity check on that data object. So that's an important thing to understand. Now, if you're going to use the package that I've suggested you to use, the Blazor Tools package, I think it's got some helpful things that will sort of make your life a little bit easier. I've got a sort of a shortcut function that will report these data in a different format. So you might recognize this now as a tibble. Essentially, it's the same thing. It's just in a different format. And it's a little bit easier to work with when we're doing manipulations with the tidyverse packages. Okay. So gene metadata. Same idea. I'm going to type in this command row data, and that's going to give us all of the information about each gene. This is a data frame with 36,000 rows and 7 columns. And as you can see here, each row is a unique gene ID. Okay, and we have a similar function here to get the data in a tibble format, same data, just in a different format. Okay. The other thing that we can do is, as I mentioned, reduced dimensions are stored within this data object. They help us understand the, the high dimensional nature of these cells. So those are stored in there, and you can pull those out. Those are the PCAs. And so you can see here, each cell has, in this case, 50 principal components, and each cell has 2 UMAP dimensions. You can see here. We don't often work with these sorry, I'm trying to find the top here. We don't often work with these dimensions in their raw format, but this is what's going to give us our XY dimensions when we make the UMAP plots. So this is also stored within those data objects. Okay, so now how are we going to generate all this? So let me just go back to the course here. So I've gone through that. Oh, and I wanted to say another thing about the sort of the framework that we're using. Most of the data structures and the functions that I use are based on the Monocle 3 package. Most of you, or many of you, may be familiar with the Seurat package, and I think there's advantages and disadvantages of both, and there's features and functions that I use from both. For example, that aren't available in the other. So, for example, I like the gene modules in the pseudotime, which are available from Monocle, but not Seurat. Conversely, making cell annotations is, I think, much better in Seurat compared to Monocle. So you kind of have to pick one that you're going to want to work with. I think the advantage of the Monocle program and package is that the data structures are simpler. They're smaller, meaning that they store less redundant data within themselves, and I think they're easier to work with. The other thing is that they're based directly on the sort of the bioconductor data structures, and so yes, there's a lot of things that work with Seurat, but there's also, I think, probably more structures that sort of functions that work with these bioconductor-based data structures. So, most of what I do is based on Monocle, but Seurat also has some things that we're going to work with. Okay, so as I mentioned, we can describe cells by number of features. And these features, the easiest features that we can think of are the things that we already know about the sample that they came from. So in this case, we're going to import this information right at the beginning. And we're going to use that table that we're going to import to set up our analysis and set up our data structure as we build it. So let me show you how that's done. Okay, so this command here is going to read in what I call a configuration file. So we're going to name it analysis configs. Okay, and let's sort of explore what is in this particular file. All right, so what we've read in here is a tibble, all right, and it's telling us about these samples. I have two samples. One is called Chromium Control, the other one is called Chromium X. Both were acquired on the same date. There's another value here, equipment. So it was acquired on a 10X Chromium. Another one was called on a Chromium X. And this path here is the path to where the data is stored. Okay, so what's the point of this data set? Well, this is essentially a very small data set that 10X has made available for us to compare a similar sample run on two different machines. Okay, so it doesn't matter what it is. We're just going to use this as an example. Okay, so that's all we know. But if you're running, like, say, a patient sample or a set of patient samples, there's potentially a lot of information that you could put in here, and frankly, it's easier to include it initially than to go in and add it after the fact. So any sample information I'm going to be putting in upfront like this. Okay. Next thing you're going to want to do is you're going to want to make sure that these paths are done correctly. You may not recognize this, but when I see these sort of backslashes, I recognize this as being sort of a problem. Oftentimes, translating from like a Windows-based file to Linux or a Mac, there's going to be problems identifying these file paths. So I have a function that's going to fix some of that, but that may not work for you. But you're going to want to make sure that this file path points directly at the folder that is holding your data. So, for example, this folder here. All right, I'll put it down like that. So that's folder here is holding this filtered feature barcode matrix.tar.gz. That's what you want to point it to. All right. So what I can do is I will fix that, and so now it's pointing to the correct file path. If you're working on your own, that is probably not going to work for you. So you'll have to just make sure that that's corrected manually. And you would do that by basically editing that CSV file so that it's correct. Okay, so we have the analysis configuration file read in. And now is when you're going to start to do some of the processing. So I'm going to walk you through this. I'm not actually going to run this. But what I'm using, you can find in the Blazor templates package. All right, and this is really easy for you to just like extract using this command if you want, and then you're going to have to do some modifications on it. The reason it's not sort of like wrapped up into a easy to use function is that it's really not possible to predict all the different sort of small changes you're going to want to make to your analysis. So you might want to change things. 10x might change things in terms of how they're providing the results to you. So this is a sort of a starting point that you're going to have to work through. You're going to encounter errors. Things aren't going to run the first time. So you're going to have to tweak it in order to make it work properly. But we're going to walk through it. And usually, at the beginning, as I indicated, you're going to read in the configuration file. I'm going to skip this. This is essentially a long sort of command here to summarize and plot, or basically summarize the sequencing metrics. You don't need to do that. That's actually provided for you in that web file that I showed. All right, so the next thing you're going to do here is you're going to generate a list of cell data sets. Okay, so we know what a list is. We know what a cell data set is. So this is going to be a list where each object in the list represents one sample. Okay, so it's going to map across the list of samples from your analysis configs. Okay, so in this case, we have 2. And it's going to then find that filtered feature barcode matrix file that I showed you. And then what it's going to do is it's going to copy those files to a temporary directory, and it's going to load those into a cell data set. And it's going to return that cell data set. This statement here, it's an if statement. So we discussed that last week. What it's going to do is if you've done any sort of multimodal analysis like scite-seq, it's going to remove those initially because you're not going to want to have those mixed in with your gene counts. Okay, so that's what that's for. That bit there is going to set the names on each of those components of that CDS list with the name of your sample. Okay, so that's pretty quick. Now the next thing you're going to want to do is to do some quality control. Now, I think this is one of the things, one of the advantages that you get out of running this type of analysis on your own, rather than simply using the raw output from Cell Ranger, the 10x genomics program, so that you can do some filtering to get rid of either poor quality cells or doublets, these sorts of things, which are things that you don't want to necessarily include. And so I've written a function that will do that for you. And what it does is it goes through each of those component cell data sets from the CDS list, and it will calculate for each cell in there the number of features that are detected, the number of genes essentially that are detected, and it will calculate the percent of those genes for each cell that are mapped to, say, mitochondrial genes. Basically mitochondrial genes. And mitochondrial genes are an indicator of poor quality cells. So we want to throw out cells that have too many reads mapping to mitochondrial genes, and we want to throw out cells that have low numbers of feature counts, because those are also poor quality. And so what this function does is going to return a list of all that useful information. And let me walk you through what it has returned for this CDS that we're working with. Okay. So the first thing that's going to list is the first thing in the list for each sample is going to give you a table of the cell barcode. and it's going to tell you whether it's been filtered or flagged by any of those QC metrics. Okay, so if it's false, that means that's a good cell. We're going to keep it. If it's true, eventually we're going to throw those cells out. All right. So item 2 for each sample is basically a violin plot of all the cells that have been all the cells on the left here, and then on the right are all of the cells after filtering out the bad cells. And we're showing this, we're showing the number log, the log 10, base 10 log of the number of detected genes. Okay, so you can see here that this sample has essentially 2 major populations. That's shown here by the width of these violins. One with a log of around, say, 3.3, another of log of 3.6. And so that roughly corresponds to between, I don't know, 2,000 and 3,000 genes, something like that. So, yeah, so that's all the cells. Now we've thrown out the cells that have this lower number of gene counts. Okay, so that's that. The other thing to show here is the percent mitochondrial reads. And here we're showing that above in mitochondrial percent mitochondrial reads of 13.2%, we are getting rid of all those cells essentially. So anything more than that is a poor quality cell that we want to get rid of. Now you can return what these values are. So the fifth item in here is the number of genes. This is the threshold, the lower threshold. So I'm going to go back to this. Actually, I'm going to go to this one here. So this is the threshold here that we've used to cut off the number of genes detected. That's 910. So cells with less than 910 genes detected, we're going to get rid of. And likewise, if we run the 6th entry here, so cells with more than 13.29% mitochondrial reads we've gotten rid of. So that's a good indicator for yourself, the quality of your sample. So 13 is, pretty high. If it was more than say 15 or 20, certainly you might question whether that sample is good enough to use. So usually it's nice to have that less than 10 or so, 10%. In any event, so that's the quality control. Now let's go back to this here. Now, the next thing we want to do is we want to try and get rid of doublets. So, so why did doublets happen? Well, it has to do with the technology that's used for this assay. And, the way it works is that the rate of doublets, which is cells that we, intended to capture as single cells, but instead what happened was that we captured 2 cells in the little barcoded cell droplet, 2 cells or even more cells. That rate is dependent upon the number of cells, the overall number of cells that we capture. So if we capture, let's say, 1,000 cells, that rate's going to be around 0.5 to 1%. If we capture 10,000 cells, that rate's going to be between 5 and 10%. So the rate scales like the number of cells that you've captured. So this formula here will calculate the anticipated doublet rate for your samples. All right. And then what we want to do is we want to feed that information into another program, which is this here called Doublet Finder. And what this does is it goes through and it bioinformatically predicts cells that are likely to be doublets. And it uses that anticipated doublet rate to help identify those. This particular function takes a while to run. It might occupy your laptop for several hours, depending upon the size of the data set. So just beware, and that might be something you need to have some help or a more powerful computer to run. But in any event, we've gone through all of the the code that you see here. So this is PMAP, so we're mapping across 3 different things here. So the list, the CDS list, the doublet rate for each of those samples in the list, and the QC calls. So the QC calls are this value here, this table here. That's what gets fed in to predict the doublets only for the good cells. All right. So that is going to return, I don't have it here, but it's going to return a table with high or low predicted probability of being a doublet. Okay. So now you've got quality control information, you've got doublet information. We need to rejoin all that information back into the individual cell data set objects. And that's what this does here. So it's going to take that list, it's going to take the QC data, it's going to take the doublet data, and it's going to stick those back onto the cell metadata. So if I run this guy again here, cell metadata, what it's going to do is it's going to put some extra columns on here that says, is it a doublet? Is it a high confidence doublet? Yes, no. Is it a low confidence doublet? Yes, no. Did it pass all of the QC? Yes, no. So that's what you get there. That's a pretty quick one. And then you're going to merge all these. So remember, we're holding all these data together in a list. We want to put them in a single object that we can do some more interesting things with. We want to analyze all the data together, obviously. So that's what that function does. The next step that I do here, so you'll recognize these brackets. We talked about brackets when we talked about data frames and essentially subsetting. Okay, a single bracket is a subset. So here you're taking a subset of the main CDS that we just made, and you're going to get rid of, okay, that not in. So we're going to take out all the genes that that are in this list of genes here. All right. So what are these genes? These are mitochondrial and ribosomal genes that are not helpful for us usually. Now you can leave them in if you want, and that's going, all you have to do is just skip that or delete that from your script. But usually these things don't help you, so we're going to take those out. So that's a subsetting function there. Okay. This function here, another subsetting function, where we're going to get rid of the low quality cells. And here we're going to get rid of the basically the doublets. So here we're selecting only the singlets. Okay. So that's how we essentially merge together multiple data sets and clean up our data. These commands here get rid of those quality control and doublet information columns from the cell metadata because we don't need those once we remove them. Okay, so the last few things that we're going to go through are calculating the reduced dimensions. Now there's 2 steps to this. So the first step is to calculate the PCAs. If you don't know what PCAs are, they're essentially the recalculations of the most informative genes that distinguish from the distance between cells. And there's a lot of good resources that you can find, on the web, like Wikipedia, for example, is a good one to figure out how those work. You can read also the tutorials from Monaco or Surratt to learn more about those things. But what preprocessed CDS does is it reduces the 15 to 20 to 30,000 genes down to, say, 30 to 50 principal component dimensions. Now, that's only operating, as you can see here, on the gene expression data. It's not going to operate on any other data like site-seq data or ATAC data. So this is strictly based on the gene expression counts. The UMAP calculations are going to be made by this command here. That's going to operate directly on the PCAs that you calculated there. All right, so now you've gone from, say, 30,000 genes to 30 to 50 PCAs. down to 2 UMAP dimensions. All right. So let me show you what that is actually going to look like. So I go back down here. And so this is the CDS that I'm using for the example. It's named differently from what I just showed you in the script, but that shouldn't be too confusing. And we can use this command here to highlight each sample in our data. Okay, so I run that, and it gives us the UMAP dimensions here. And each, so there's UMAP 1, UMAP 2, and each sample is labeled here by a different color. All right. So I think that's probably a good point to stop. And I'm going to wait here and see if anyone has any questions about what I've gone through. I know there's a lot. It's pretty complicated, but you should be able to get those files, look at them, and work through them and understand how they're operating. So any questions or concerns or anything else in the audience? Doesn't look like it. Okay, good. I guess it must be clear as mud. So let me cover one more thing here, which I think is important, and then we'll pick up where we left off for last week, we'll pick up next week where we left off this week. So one thing that comes up occasionally is batch effect. Now, you want to try to avoid that if you can, but sometimes it's unavoidable, and you'll have to repeat experiment on several different days, different runs, these sorts of things. And this can induce some unwanted variability in your data. And we can correct for that using this command here. And what it does is it essentially will take those PCA dimensions that we had calculated, and it will recalculate them by aligning sort of the k-means. It will find like the center of groups of cells, and it will align those and recalculate the dimensions based on a variable that you put in. So in this case, it says here batch. So let's say you had 2 or 3 samples in each batch. It will find the centers of groups of cells based on those PCAs according to batch, and then we'll calculate an equation to align those together, and then it will regenerate the PCAs based on those equations. And this is a very useful feature if you're seeing batch effect in your data. Now, in this case, there really is no batch effect, and we don't have batches to work with. So I can't show you anything how well this works, but in this example data set, I did run that alignment procedure. And the point that I want to make is that we don't, and when you run that with the program that I showed you, it will preserve the original dimensions for you. So if I run this, these are actually the aligned, in this case, UMAP dimensions. And then if I run this, I have these alternate dimensions that we're storing in this data object. You can even see that this is now changed. It's now pre-alignment dimensions. So if you're doing that, you don't worry that you're losing your data, or that that you can't go back. So that will be maintained in there. So yeah, like I said, there was no significant difference, obviously, between these samples. They overlap nicely. So it's not something you would have needed to do. I just am showing you as an example. Another important thing to point out about alignment to correct batch effect is that this procedure only operates on the reduced dimensions. It doesn't change the actual gene counts for the cells. So if you go through and you say, all right, now I've done all this alignment, now show me the differential expression between, say that cluster and that cluster, those batch effects are still going to be in the data. So you have to then correct for that again later on. And so you have to be aware that that's an issue in your data. Otherwise, you're going to have unwanted variability in your downstream calculations if you don't also correct for batch doing those things. Okay, so I think we can call it a day now. And I want you guys, if you can, to try and run through some of these commands here, not the processing commands, but some of the commands that are on this week 4 and 5 script. This will be pushed up to GitHub, so you should be able to see it. You should be able to, copy it, paste it into your project. And if you load these packages, you should have access to this vignette CDS, and you should be able to run everything. So I'd like you guys to try and run through those things. Come up with any questions, any problems, put them in the issues section or email them to me. And hopefully, you'll be sort of on your way to working with your own data sooner rather than later. All right, so we'll call it a day. And thanks again, everyone. And we'll see you next week. Bye.
Thank you.
You're welcome.
Bye.
Bye.